Hello class. During week 3's lecture, we finished by talking about the low GC content bacteria. It's very important to note that, again, when we're talking about GC content, we're talking about the guanosine, cytosine content. So we're talking about the percentage of those bases as compared to the adenosine and thymidine bases in the genetic material. Now, again, as I mentioned before, we finished our discussion in week 3's lecture with the low GC content bacteria. So where I'm going to pick up in this video recording is with the high GC content bacteria. And hopefully you remember from last week's lecture that this simply means that now we're looking at bacteria with a higher, at least 50% or more, guanine, cytosine percentage of bases as compared to adenine and thymine. These organisms all belong to phylum Actinobacteria, and they are commonly found in soil, fresh water, as well as in marine environment. All of them also, or most of them, play a role in the decomposition of organic materials. So we'll talk about some of those environmentally important bacteria, and we'll talk about those that are considered to be pathogens. And because they are pathogens, that means that they cause disease. As I mentioned during week 3's lecture, your textbook contains plenty of genera that are listed. We're only going to focus on a select few, and those would be the ones that you are responsible for. So I will be covering these bacteria, but I'll also be covering others as well. Again, take note of this table in your textbook, table 11.2. With the high GC content bacteria, now we're looking mostly at those that belong to phylum Actinobacteria. So the Corynebacterium will be next, Corynebacterium. These are facultative anaerobic organisms. They do happen to be gram-positive. They include strains of the pleomorphic organisms. So, as I mentioned before, when they are pleomorphic, that means that they have abstract cell morphologies. They also happen to be aerobic, so we know now that they require oxygen in order to survive, and therefore they go through aerobic respiration. Also, we have dipterial as well as non-dipterial strains. This simply refers to the pathology that they lead to, and as such, the toxins that they actually produce. So in regards to this bacterium, it leads to localized inflammation, usually in the upper respiratory tract, and it leads to the condition known as diphtheria. This is for the diphtherial strains. C. diphtheriae is the causative agent, and it produces this particular toxin here, which would be the diphtherial toxin. This simply prevents polypeptide synthesis from occurring, and it leads to cell death. This term, polypeptide synthesis, refers to protein synthesis. Diphtheria is categorized by a sore throat, low fever, and an adherent membrane. So this is a specific symptom here that this organism leads to. An adherent membrane, we also refer to it as a pseudomembrane as well. This will occur on the tonsils, pharynx, and within the nasal cavity. A milder form can be restricted to the skin, in which case it will still be considered diphtherial. In less common cases, in about 20% of the cases, myocarditis can also occur as well, and peripheral neuropathy. Now, for those belonging to the Mycobacterium genus, these are aerobic as well as non-motile rods, and they sometimes form filaments. So by filaments, that means that if we were to look at them under the microscope, they will have a filamentous or more of an elongated shape. These organisms all contain mycolic acid, and hopefully you remember from lecture 2, from week 2's lecture, that mycolic acid is a waxy lipid that's present within the cell walls of gram-positive or of select gram-positive organisms, and this happens to be the case with this particular genus. Due to the fact that they have mycolic acid, they grow very slowly as well, and this usually is the case when someone has latent TB. That's due to the slow growth. So Mycobacterium tuberculosis will be the most common causative agent. This one remains viable for a long time in aerosol droplets. So those are simply droplets that we can inhale that contain the bacteria. As someone sneezes or coughs and they are infected with this bacterium, if it's actively growing, they can shed it within the droplets, and then once we inhale those droplets, that's how it becomes transmitted to us. So when it says viable here, this simply means that the organisms remain alive. There are 3 major types of tuberculosis, and they are primary, secondary, and then disseminated. Symptoms are not always initially apparent, and again, this is due to the fact that the organism grows very slowly in the beginning stages. So if it's growing very slowly due to the mycolic acid, then the symptoms may not be as readily apparent, because at that point the patient doesn't have large numbers of the bacteria present in the body. However, in regards to when the symptoms appear, this really is determined by not only the microbial load of the organisms that are present, and in other words, microbial load equates to the amount of bacteria that are present, but also whether or not the patient is immunocompromised. Individuals that are immunocompromised, such as those with HIV, are most at risk. And it happens to be the leading cause of HIV positive individuals, and it is an opportunistic infection within HIV positive individuals. So we can diagnose it by performing a tuberculin skin test. Most of us have had those to enter into school or to obtain certain types of jobs. This actually identifies exposure to the bacterium that causes tuberculosis. At that point, if the tuberculin skin test is positive, chest x-rays will next be used to identify tubercles within the lungs. And then treatment actually requires a combination of drugs. A lot of strains of M. tuberculosis are becoming multi-drug resistant. So to counter that resistance, drugs are given in cocktails. Also, due to the slow growth of the organisms, drugs are given in cocktails and for longer doses as well, or for longer periods of time as well. And as you can see in the last bullet point, the BCG vaccine is available where tuberculosis is common. So what you're seeing on this slide is the inhalation of droplets. Again, the organism is shed via aerosol droplets. Once we inhale those droplets, they enter into the lungs and into the alveoli. At that point, you will have macrophages that will go to the site and engulf those organisms. However, these organisms are able to survive within the white blood cells. That's what you're seeing here. As the infection increases, you will have more macrophages that are recruited to the site, and this is the beginning of a tubercle, or the formation of a tubercle. At that point, as it becomes more and more advanced, and this stage 4 here, what we have is primary tuberculosis infection. As it becomes more extensive of an infection, now what you have is more tissue damage, at which point it can rupture, it can cause necrosis, and now that can lead to secondary or reactivated tuberculosis. What you're seeing here is a picture showing a positive tuberculin skin test, after which point, again, a chest x-ray will be performed to look for the presence of the tubercles. Those are the Nocardia and Rhodococcus genera are next. These two, very similarly to the Mycobacterium genus of bacteria, have mycolic acid in their cell walls. So we would not perform the Gram staining technique with Mycobacterium nor these two genus, even though we know, or these two genera, even though we know that they are Gram positive from other types of analyses that we can perform. We would actually perform an acid-fast staining test to determine whether or not these organisms are what we say are acid-fast positive. And what that simply means is that if they are acid-fast positive, that means that they have mycolic acid present within their cell walls. For the Nocardia genus, they are gram-positive. Again, they are rod-shaped organisms. They are non-pathogenic as well as pathogenic forms, and then most infections are acquired by inhalation or through traumatic injury. Very similar to what we were talking about earlier in the week Traumatic injury simply means that there is some sort of trauma that introduces the bacterium into the body. Usually those that acquire these infections are immunocompromised, and as such, nocardial infections are considered to be opportunistic, simply meaning that they take advantage of the opportunity that now someone has compromising of their immune system. For the Rhodococcus genus, they are aerobic, non-sporulating. So here, non-sporulating simply means that the organisms do not produce endospores. You all should remember that simply because only those bacteria of the Bacillus and Clostridium genus can turn into endospores. They too are also non-motile and they're gram-positive as well. Very few species are pathogenic. Most are benign, meaning that they're non-disease causing, unless, of course, again, the individuals are immunocompromised, in which case they become opportunistic pathogens. The next phylum will be the Proteobacteria phylum. This happens to be the largest as well as the most diverse phylum of the eubacteria under domain bacteria. All of these are going to be gram-negative, and they have many extensions including prostheticae, which are basically cellular extensions that are used for increased surface area to obtain more nutrients. So these are not cellular extensions that are used for motility. They are not utilized for motility. So a wide variety of pathogens are included under this particular phylum, included Escherichia, which is the genus to which E. coli belongs. There's also the Salmonella genus, Vibrio, and Helicobacter, which is the subject of the case study that you all received for homework. So the Proteobacteria are all divided into 6 sections, and they are referred to by Greek letters starting with alpha all the way through zeta, and they're listed here. So these are just the subcategories of phylum Proteobacteria. They include alpha Proteobacterium, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, epsilon Proteobacteria, and zeta Proteobacteria. We denote these different categories based upon their ribosomal RNA content. So what you're seeing here is the prosthetica. Again, this is utilized for increased surface area so that they can obtain more nutrients. This is not utilized for motility. So table 11.3 summarizes all of the subcategories of this particular phylum. Again, starting with alpha, going all the way to zeta. There are 3 listed here, alpha, beta, and gamma. You're looking at some representative genera here within this group, special characteristics, and then some diseases. Again, you're only responsible for those genera that I mentioned. This portion of the table just continues with the delta and epsilon Proteobacteria. Again, representative genera here. Okay, so a lot of these are involved in environmental cycles. So first we'll start with the alpha Proteobacteria, which will include a nitrogen fixers such as Azospirillum and Rhizobium. They are commonly responsible for tiny nodules that appear on the roots of different types of agricultural plants such as soybeans. They are responsible for converting nitrogen to ammonia gas. Also under alpha Proteobacteria will be the Rickettsia, Brucella, and Ehrlichia. With Rickettsia, this is transmitted by many ticks, fleas, and lice. When we have bacteria that are transmitted via insects, which are commonly found on animals, these are referred to by the term zoonoses, simply meaning that we can now acquire these from animals. So zoonoses. So Rickettsia causes many diseases in humans, such as typhus, rickettsiopox, African tick bite fever, and then also Rocky Mountain spotted fever. Again, the major way they are transmitted are via these insects, so ticks, fleas, and lice. And these are what we would consider to be organisms that are blood-sucking arthropods, meaning that they're able to burrow into our skin or bite into our tissues and deposit the organisms there. Next is brucella. These are transmitted among animals. So again, these will be referred to as zoonoses. So some example of animals here would be sheep, goats, cattle, deer, elk, and pigs, as well as dogs. Humans can become infected with them by coming into contact with the animals or the animal products. They are contaminated with these bacteria. So whether we're eating the animal specifically or if we're coming into contact with their waste products, and if we're not washing our hands properly, we can actually self-inoculate. Brucella leads to brucellosis, and this is a disease that is characterized by spontaneous abortions. It can also lead to sterility if not treated properly. And they are able to survive phagocytosis by our white blood cells, and as such, they are considered to be intracellular parasites. Ehrlichia is also transmitted by ticks as well. And there are several different species that can cause a variety of diseases in humans, such as ehrlichiosis. These are also intracellular parasites, and as such, as you can see here, they're able to live within white blood cells. Okay, next is Bordetella pertussis. We commonly just simply refer to this as pertussis. This leads to whooping cough. Initially, infected individuals will have cold-like symptoms, and then they will develop the characteristic cough, the whooping sound. So there are numerous virulence factors that are produced by this organism, including adhesions, which are proteins that allow for it to adhere to the epithelial of the host. And then they also produce toxins as well. Our immune system launches responses to these toxins, and as such, that's what leads to the characteristic symptoms there. Pertussis does progress through each of these 4 phases. And once convalescence is achieved, that means that they no longer have the signs and symptoms. This is a highly contagious condition. The bacteria can spread through the air in airborne droplets. And again, the symptoms, which will be the whooping sound of the cough, are usually diagnostic. Treatment will be primarily supportive, and the best way to prevent it is with the DTaP vaccine. So if you know you will be working with young children, particularly infants and toddlers, it is recommended to get the DTaP vaccine. The gamma proteobacteria group is next. This includes the Pseudomonas, such as Pseudomonas aeruginosa. This is a gram-negative bacteria that grows a yellowish-green color on agar, so you'll notice in lab whenever we are culturing Pseudomonas, it will cause the agar to turn this color. These are commonly the causes of urinary tract infections and swimmer's ear, and they are occasionally found in the anogenital and axilla areas of the normal skin. Rarely, however, do we see them shed via fecal matter. Pseudomonas infections can develop in the subcutaneous tissue, as well as the skin, ears, eyes, and bones, as well as urinary tract, and then heart valves. So for each of these locations that are mentioned, the site infected actually depends on the mode of entry. Mode of entry simply means how does one acquire, or at what site does one acquire the organism, and how is it acquired? Is it via, again, traumatic injury, self-inoculation, so on and so forth. Pseudomonas does happen to be an opportunistic pathogen, so all that means is that it takes advantage of the opportunity to infect an individual if they are immunocompromised or suppressed. Pseudomonas happens to also be the causative agent of a lot of nosocomial infection. This term nosocomial infection simply means that the organism is acquired in a healthcare setting. So here are some pictures here that shows you how Pseudomonas infections look. And you can see the characteristic yellowish green color arising from these infections. Escherichia coli, we commonly refer to this organism as E. coli. That's the next bacterium. They are gram-negative, rod-shaped bacteria. They are commonly found as a part of our normal flora in the lower intestine of warm-blooded organisms, again, such as humans. Most strains are harmless. For those that, again, are considered a part of our normal flora, they are going to be harmless as long as they remain in the areas of the body where they should normally be found. So again, the lower respiratory tract. However, there are many different serotypes, which again, I told you this week, simply means different strains of the organism. And depending upon the serotype that someone becomes infected with, that can then lead to other types of conditions such as serious cases of food poisoning. Occasionally, they are also responsible for food recalls and product recalls. Harmless strains, so those that are considered to be a part of our normal flora, actually benefit us. So they're actually mutualistic bacteria in that they actually produce vitamin K, which is essential to proper digestion. Now, when we get other serotypes or other strains that are more harmful and therefore more virulent, that's when we can begin to develop infections. So in regards to our normal flora, Escherichia coli constitutes about 0.1% of gut flora. The fecal-oral route is the major route of transmission. So this is a very key point here, a fecal-oral route. When we say fecal-oral route, what that simply means is that the organism has been transmitted via the spread of the fecal matter. So this can come about, for instance, if you were infected or if you had this particular organism present, just as a part of your normal flora, and if you do not use or if you do not wash your hands properly after using the restroom. You can then transfer it to other areas of the body. Specifically, since it's a fecal-oral route, it will be back into the mouth. So if you were to prepare food without washing your hands properly, and you eat that contaminated food that now has the fecal E. coli that you shed, that's considered to be the fecal-oral route. And this is actually the major route of transmission for most microbes that we acquire. So that's considered to be the #1 mode of entry. In regards to E. coli, the cells are able to survive outside the body for a limited amount of time, and they're ideal organisms to test when we're looking for fecal contamination. Now, there are virulent strains, and the one that you see here on the board, the E. coli 0157H7 strain, happens to be a highly virulent strain. So this one is also referred to as enterohemorrhagic E. coli or EHEC. Now, this particular strain of E. coli is different from the ones that we normally have as a part of our normal flora. Again, the ones that are a part of our normal flora reside within our bodies without normally causing harm. However, when we come into contact with the 0157H7 strain, it can lead to bloody diarrhea, severe anemia, hemolytic uremic syndrome, and kidney failure, all of which can lead to death. For a healthy adult that's not immunocompromised or immunosuppressed, they will usually recover from an 0157H7 strain within a week. If they are immunocompromised or suppressed, however, they will suffer from the aforementioned symptoms. The same holds true for other demographic groups, such as the really elderly and the really young. So this can prove fatal in select groups. Salmonella is the next genus. So these two are rod-shaped and gram-negative. They are non-spore-forming enterobacteria. So now when you see this term enterobacteria, that means that these bacteria are present within the gastrointestinal tract, and they can impact the gastrointestinal tract. Similarly to E. coli, there are many serotypes, so there are many different strains of Salmonella. The two more common types would be S. typhi, Salmonella typhi, and then Salmonella enterica. These are motile organisms. They do have flagella that grade in all directions. So what that simply means is that they have the peritrichous format. So if you recall from about week 2, The peritrichous format would be when the flagella are all around the cell body, pretty similar to what I've drawn on the board. Now these infections are zoonotic. So zoonotic goes back to the term that I wrote earlier, which would be zoonoses. What that simply means is that they're transmitted between humans and animals. So I told you guys a little earlier in the term about the recent recall or the recent scare that we had with chicken that was contaminated with salmonella. So again, when we obtain the organism from animal products that are contaminated, that's considered to be via zoonotic means or through animal means. So many human infections from salmonella are due to the fact that we are ingesting contaminated food, such as chicken. Salmonellosis is the condition that it leads to. These organisms, the Salmonella genus, is actually closely related to Escherichia genus as well. They are found worldwide in cold as well as warm-blooded animals, including humans, obviously. And they can cause illnesses such as typhoid fever, paratyphoid fever, and foodborne illness. The condition that results is dependent upon the serotype and the species of organism that one is contaminated with. They primarily enter in via the oral route, and that's through the digestive tract. Therefore, they must be ingested. However, they must be ingested in large enough numbers to cause disease in healthy adults. Otherwise, our immune system will eliminate them with relatively little harm to us. So again, as I've been mentioning before, for individuals that are immunocompromised or immunosuppressed, they are most at risk. Also, gastric acidity is responsible for the destruction of most of the ingested bacteria that we would come into contact with, even if we were to eat contaminated meat. So that's one reason why it doesn't necessarily cause disease in healthy adults, because these are more neutrophilic organisms. Therefore, the acidic environment of the stomach will kill them. And then in addition to that, a lot of these bacteria may become trapped in the mucus that's produced in the esophagus. So we do have a lot of protective mechanisms within the body that serve to protect us from these potentially pathogenic organisms. The problem comes, however, when one is immunocompromised or immunosuppressed, or again, if they are ingesting large numbers of these organisms that our immune system cannot combat in an efficient manner. Helicobacter pylori is next. This one is commonly referred to as H. pylori. These are gram-negative bacteria. They have a helical shape, hence their name. They are microaerophilic. If you recall from last week's lecture, from week 2's lecture, I mentioned that microaerophilic organisms are those that only require large quantities of, small quantities of oxygen, and they are found in the stomach. Therefore, H. pylori is considered to be an acidophilic organism. It requires an acidic environment. They were first identified in 1982 in patients with chronic gastritis and gastric ulcers, both of which were not previously believed to be caused by microbes. However, as the correlation became stronger, scientists were able to finally determine that this particular genus and species of bacteria were responsible for gastric ulcers. Statistically, we now know that more than 50% of the world's population harbor H. pylori just naturally in their upper respiratory tract. However, in regards, again, to more epidemiology, the infection, broader H. pylori, is more prevalent in developing countries, and the incidence is actually decreasing in Western countries as we know more about how to control this organism. The bacterioids are next. This is a genus of gram-negative bacteria. They are bacillus. They're also non-endospore forming. They're anaerobic organisms, and they can be either modal or non-modal, depending on the species. Normally, these organisms are also a part of our normal flora, therefore they're mutualistic, and they make up a substantial portion of our microbiota, so the normal bacteria that we would normally have living within the body. They also help us to process complex macromolecules into more simpler molecules that we can metabolize. Vibrio is next. This is a genus of gram-negative bacteria. They are comma-shaped, hence the name Vibrio. Some strains, such as Vibrio cholerae, cause the disease cholera. These are facultative anaerobic organisms, and it has a single flagellum at one polar end of the cell. In regards to cholera, it is transmitted primarily by drinking water or eating food that has been contaminated by the fecal matter of an infected person. As such, individuals that are asymptomatic can also transmit the bacteria. The severity of the diarrhea, as well as the vomiting that arises from a Vibrio cholerae infection, can lead to rapid dehydration, which then leads to improper electrolyte balance, and ultimately, if this is not corrected, death will ensue. The primary treatment for cholera will be oral rehydration therapy, and that's to replace the water as well as electrolytes. Hopefully, as you guys know from your previous classes, without proper electrolyte balance or water, we cannot metabolize. And if we can't go through metabolism, we will die. So that's why it's very important to continue to rehydrate these individuals. Without proper hydration, it can lead to failure of the kidneys. And at that point, that can lead to multi-organ failure. So in regards to the epidemiology of cholera, it affects about 3 to 5 million people worldwide and causes approximately 100,000 to 130,000 deaths each year. These were the statistics as of 2010. Next is Legionnaire's disease. And we talked a little bit about Legionnaire's disease last week. You had a case study on it when we were talking about biofilms. So Legionnaire's disease is caused by the organism Legionella. This is a gram-negative bacteria. Again, it falls under the Proteobacterium or Proteobacteria phylum. It was actually named in 1976 because of the first outbreak, which occurred at a Legionnaire's, an American Legion conference in Philadelphia. Legionella does survive in domestic water sources, so as I was mentioning during the case study activity last week, it can contaminate plumbing and piping systems, and as such, when we come into contact with it, it can lead to this particular condition. So in regards to those that are primarily impacted, it will be the elderly, as well as smokers and immunocompromised individuals. They are most at risk. The infection is very similar to pneumonia,